Pronto, pronto? Mi sentite?
Simona, allora, ma quale dei due? Perché io sono entrata in quella con gli studenti e ora entro anche in quell'altra. Aspetta.
Questa è per tutti, penso.
Quella lì sì, anch'io ci sono. Ma quell'altro, quella solo per la commissione invece?
Non lo so, ora glielo chiediamo,
Aspetta, c'è. Sì, ancora però non accedi dal browser, vediamo questo qua. Ah, ok, adesso c'è anche questa. Ok, vai. Come? Non ho capito, Simona. Simona? Eh. Nel calendario ce ne sono due indirizzi.
Ah, io ci sarò, però ci sono poco d'accesso da di là.
Sì, anch'io, ma ora funziona anche quell'altra.
Quindi basta che ci clicco sopra.
Prova, io adesso Lorenzo Bellani, boh, non è in corso, però per adesso ancora.
Io join.
Sì, però io ancora non vedo nessuno. Non è in corso, cioè quella degli studenti vedo Davide De Rossi, la faccia di Antonio, Rohit, però non è che. Però vabbè, sono le 4 e un quarto, vediamo.
Ci farà Lorenza Belloni.
Ah, io
Si
però.
è fatto i capelli,
Sì, hai visto? Sono andata a tagliare i capelli. Però te non ti vedo, dove sei? Aspetta, non ti.
Sta carina.
Ah, eccoti, eccoti. Ti vedo qui, ma di qua non ti vedo. Vabbè. Andrea Cresti è comparso ora. Eccolo.
Buongiorno.
Eccolo Andrea. Ok, vai. Però si vede. No, infatti.
Quindi hai cliccato quella a destra.
Sono tornata indietro su quell'altro link solo commissioni. Quindi io ce n'ho due aperte adesso praticamente. Eh. Aspetta. Boh. Quindi questa è quella con gli studenti, quell'altra. Oh, vai. Non riesco. Allora, interrompi video, disattiva. Interrompi video vuol dire spengere la telecamera, no? Sì. Ah, ecco Fabrizio. Vai, ti saluto. Vai, ciao ciao. Ciao Fabrizio.
Ho visto il Carapelli in icona, sembrava vivo.
Aspetta. No, Simona, sento un gran fischio. Ah! E poi non ti vedo, Simona, qua. Non ti vedo nell'altra pagina, nell'altra... E qua non ti sento. Fai qualcosa.
Che eleganza. Giustamente, giustamente, bravo. Anzi, ora mi assetto anch'io.
No, come mai? Il microfono. Cos'è?
Buonasera.
Buonasera.
Dicevo, le dobbiamo tenere aperte tutte e due le sessioni?
Io ho solo questa.
Perché quella in
Welcome
streaming
everybody.
che ci si fa? Quello sarà per il pubblico.
Welcome, welcome, welcome.
Con-
Webcam? Webcam?
And webcam.
No, perché con Webex non si capisce bene.
Ma infatti perché no Google Meet non è più...
Perché ci sono i disturbatori,
Intanto niente.
quelli che poi se a metà sessione di laurea ti sparano il porno, come è successo alla Marzili quando faceva la presentazione.
Stefano, io non ti sento.
Okay, it was a long day. I started with the graduation day early this morning, and the main reason why they have skipped to the Cisco WebEx system is that it was not possible to broadcast the YouTube streaming that is apparently necessary for each graduation. So that's the main reason why we are using this new device. And excuse me very in advance, because the system is really very much complicated. I have to activate the YouTube streaming and then come back to this Cisco WebEx system. And I'm not really very familiar with all these, updated and very, how do you say, modern system of broadcasting. However, now we are connected in the main room, and the system is basically the same as in Google Meet. So there is a way to share the screen for the student to broadcast the presentation. Once we have finished all the presentations, the members of the committee will be kindly invited to migrate to another private room. So you will receive a pop-up. You agree with the pop-up, and you will be moved in a different virtual room. Excuse me for the international student, I will speak in Italian the same instruction. Dunque, una volta finita la discussione delle 3 tesi, vi arriverà solo a voi commissari e ai correlatori un messaggio pop-up che vi permetterà di accedere ad una stanza privata dove saremo solo noi. Quindi non dovete fare altro che accettare il pop-up. Lì discuteremo tutte e 3 le votazioni, per poi tornare nella stanza principale.
Antonio,
Dovete disattivare i microfoni. Chi ha i microfoni, would you please disconnect your microphone?
Antonio, io non ho capito nulla quello che tu hai detto. Io non ho capito nulla quello che tu hai detto perché c'è un eco terribile dietro.
Simona, disconnetti un attimo il microfono tuo, brava, ok. Stavo dicendo che dopo aver ascoltato le presentazioni dei 3 studenti, riceverete solo voi commissari un pop-up sullo schermo. Quindi dovete accettare questo pop-up e che vi consentirà di accedere ad una stanza virtuale privata dove decideremo il voto. Dopodiché torneremo nella stanza principale per la proclamazione. Va bene? Ricordatevi, siamo in diretta streaming su YouTube, quindi per ovvi motivi insomma, comportiamoci come dovremmo. Allora, intanto vediamo. Sì, lo streaming è acceso. Io attivo la registrazione. Right, so I will come back with English again. If there are questions, it's the time to ask for questions. Otherwise, we'll start with the session. Is everything fine with the students? I guess so. Okay, so all the committee members are connected, the student as well. So I'm very very happy to start this biodiversity and environmental health session for the master degree in biology. And the first student will be, excuse me, okay, Mr. Kawan Rohit, that is defending his thesis project that is entitled Water Footprint Analysis on Cannabis Sativa Paper Production. So, Mr. Kawan, please, you are kindly invited to read the first PDF file. Do you have it in your hands?
Yeah, thank you.
Okay, so you can start reading the content of this PDF file.
Okay.
No. Kawan, you should read the PDF file before starting the presentation, please. If you don't have it, I can show you on the screen. Wait. Rohit, do you have the file with you? Can you hear me? Do you have the PDF file? I mean the declaration file. Wait, I will help you. Hold on. Hold on. Rohan, we can hear you. We can't hear you. Wait. Rohan, you are invited to read this file. Rohan, can you see my file? Everybody is reading the file, I guess. Rohan, give me a sign you are ready. Rohan, please try to disconnect and connect again. Ma come si fa?
Yeah.
Okay. Can you hear me? we wait a few minutes before until Rowan try to connect again. Guess he has a low connection.
Antonio, proviamo a spegnere le telecamere, può darsi che lo facilitiamo.
Ma sembra un suo...
Tutti
Sembra un
noi
suo,
tranne te, insomma.
sembra un suo problema, sì, comunque proviamo. Fabrizio, te hai modo di contattarlo eventualmente?
Così no, noi, sì, in italiano. Si è fatto le prove su Meet e su questo dice era affidabile, ma
Ok. Rowan is back. Rohit Kawan is back.
Rohit,
Rohit, can you hear me? Okay, Rohit. Rohit, try to speak, please.
Hello.
Okay,
Yeah.
now I hear you. Listen to me. Listen. Before starting with the presentation, you have to read the content of this PDF file. That's mandatory. Okay. So please read this document.
Okay.
I'm going to show the document.
Okay.
Go on. Read.
Line graduation. Being to 47 slash possibly in of documents laws like is yes, the end of the recording of final set into mail address and witness
Rohit, your connection is not working. We haven't heard a single word. Can you improve the quality of your connection, ASAP, please? Rohit, we can... What? Yes, I can hear you. Who's speaking? Ah, si può fare così, via. Cioè. Rohit, we don't hear you.
Cioè, a me per messaggio non mi risponde, comunque. Passo e chiudo.
Rohit, can you hear me?
Oh, hello. Hello.
Try to
Hello.
start
Now,
with the presentation.
okay.
Share your screen.
My screen is visible.
Not yet. Wait. Yes. Now, full screen.
Okay.
The screen is fine.
Yes.
Oh,
So start with your presentation, please.
okay. Like first, I'm really sorry. I don't know what happened with the connection. So my name is Rohit Chauhan. I am a master's student in biodiversity and environmental health. My thesis topic is water footprint analysis of cannabis sativa paper production. The paper production and consumption have large effects on the environment. As per now, worldwide paper consumption has risen by 40% due to its in the last 40 years due to its lead increase the deforestation via 35%. According to Indian government data on a wastage, paper industry accounts up to 30% of total waste generated in India and nearly 40 million tons of waste produced every year in India. Currently, 88% of paper is made from a tree pulp and only 12% of paper is made from various natural fibers. One of the main important process in the paper manufacturing is the removal of lignin, which is associated with the intensive consumption of water. Here you can see one picture from a local industrial's common waste space in Jaipur. Uh, The primary source for paper manufacturing is characterized by two categories. First is wood and another is non-wood. As we compared wood with the substitute natural fiber hemp, we found numerous benefits such as short growing season, low irrigation and fertilization necessities, and low lignin concentration comparing to tree, allow us a significant reduction of water throughout the supply chain, production chain. Hemp is an annual dioecious plant. Stamp have a strongest natural fiber in the world. Many environmental benefits such as phytoremediation, carbon saturation. Hemp was used in a distant past for a paper manufacturing. In the early 19th century, hemp was the primary source for a paper manufacturing. United States of America, Declaration of Independence and Constitution both written on a hemp paper. Here you can see a picture of a wild hemp plantation in the northern part of India. The water footprint concept introduced in 2003 by the Professor Erjan Hoeksetra from the University of Twente, Netherlands. The water footprint is the quantify the total fresh water used to produce the product. It is an indicator the water used through a directly or indirect consumption to produce the product. It is calculated the quantity of fresh water used to produce the product, and it is expressed as a uh, metric cubic water to produce per ton of a product. Water footprint have three components: blue water footprint, the volume of rainwater used to produce a product. Blue water footprint, the volume of groundwater used to produce a product. And the gray water footprint, the volume of water polluted during the production chain. Cellulose is one of the main component to produce a paper due to its durability and strength. The paper manufacturers facing challenges to reduce their impact on water resources. Water is one of the key components to make a cellulose to become a paper, but no study has been done before to investigate the water consumption of hemp paper production. The primary aim of this thesis to access the water footprint of hemp paper and the benefits of hemp paper production in India and comparison of hemp paper water footprint with the tree paper water footprint. The Hussein Paper Private Limited is one and only industry in India who uses hemp fiber to produce hemp paper. It is located in the northwestern part of India in the city of Jaipur. Here you can see the production area within a factory and the drying area of factory in within a factory. Hemp paper production process. First, fiber comes in a factory. The lead is cut to small clean. After the clean fiber mixed with the water to make it a pulpy. With the help of chapari, we mold hemp pulp. When the pulp is molded, it is put on a stick on a wall and hang on a string to drying under the direct sunlight. And with the help of a camel worn to remove the small stones and dust. Uh, for the shining, we use a natural process with the help of corn flour. We make a paper shine, and for removing the unwanted fibers with the help of ackee stone done. For the production of 1 ton hemp paper, we use the bleached fiber, which is bleached with the help of hydrogen peroxide, which is less toxic than chlorine and dioxin. For making a 1 ton hemp paper, around 1400 kilogram of bleached fiber was used during a thesis. Water footprint calculation of hemp paper. For the quantification of the water footprint of hemp paper, the method of Oxectra was used. The water footprint of hemp paper is total volume of fresh water that is consumed directly or indirectly. It considering all the steps during the production from the hemp cultivation, fiber processing, and hemp paper production. For the calculation of water footprint, this formula has been used. The fiber value is used. The water footprint of hemp fiber processing and water using during the production chains is manufacturing is used. The value fraction is the ratio of the market price of the product, and the product fraction is the ratio of the output product from a input product. The company uses groundwater for their production, and on an average per day, company uses 500 liters of water to process 75 kilogram of hemp fiber. Out of 500 liters, 95 liter was used for a soaking of fiber, 150 liters for the whey extracted crushed fiber of hemp, and 200 liters during the production line, and 55 liters for the cleaning of line. According on the basis of this calculation, we used around 12.5 metric cubic water for the production of 1 ton paper in a industry in India. For the water consumption of hemp growing in a growing stage is recorded 2,566 metric cubic per ton, and out of this 1,902 for the blue water footprint, and there's no need of any irrigation. That's why the blue water footprint is 0, and the gray water footprint was recorded 644 metric cubic per ton. As I said, the product and value fraction of hemp paper. Here you can see the product fraction of paper is 0.60 and the value fraction is 0.45. This diagram is drawn on the basis of data from a Ventris 1997. The water footprint of hemp fiber processing. Most of the used during the rendering of the fiber, and 36 metric cubic per ton of water used during the bleaching of fiber. And this diagram is based on the data from the Vende Werf and Tour in 2008. Here you can see the flow chart of water footprint of hemp paper production. With the help of all these values, we receive our water footprint of hemp paper is calculated to 124 battery cubic per ton. As we found, the water footprint of hemp paper is 2124 metric cubic per ton. That is 9 times less than the conventional tree paper. On the basis of study we found from the Hall and etc. 2011. This is the only one study is done till now for the water footprint calculation of of the conventional tree paper, and for the hemp paper we did, and we compare both the studies, and we found hemp paper water footprint is 9 times less than a tree paper. Here is a comparison of water footprint of hemp paper with the other products. This data is based on the liters of consumption of water in liters per kg of product produced. So here you can see for the chocolate around 25,000 liters of water is necessary. And for the conventional tree paper, around 10,000 liters of water is necessary. And for the making of 1 kg of hemp paper, only 1,000 liters of water necessary. Here you can see the clearly difference of 9 times between conventional and hemp paper. And hemp paper production we found to have a 9 times lower than the conventional tree paper. The favorable water footprint of hemp is especially important considering that this species can grow well, can be cultivated in areas where water is scarcity. Based on the study of a small Indian factory of hemp paper, it emerged that this industry is not only a water efficient, but it is a powerful driver for economic growth and long-term sustainable development with many ecological benefits. Here you can see like a difference comparison between a hemp paper and tree paper. Hemp paper is a stronger and more durable than a tree paper. Hemp can be grows more in in a acre than a tree. Hemp can grow in a 4 to 5 months of cultivation, where trees need 15 to 40 years. Hemp paper can last hundreds of years than the tree paper. Hemp paper is a biodegradable and renewable, and hemp paper can be recycled 3 times more than a tree paper. And fewer chemicals is necessary for the production of hemp paper. Only for the bleaching during the bleaching, which we can achieve with the hydrogen peroxide, that is very less toxic and than a chlorine and dioxin. Thank you for your attention. And here you can see some pictures during the thesis. And if you have any questions, please.
Yes, thank you, Rohit. And now is time for questions. So the first examiners is Professor Lopi, that I would like to see on the screen before starting for question. Please, Stefano.
So Rohit, thank you for your nice and interesting presentation.
Thank you so much, Professor.
I have to admit that I've never seen, I think at least, I've never seen a hemp paper, so I've never checked how is this kind of paper.
Okay.
My fault. Maybe I will have the chance in the future. You have shown clearly that there are great advantages in using hemp paper compared to conventional wood paper. But what I'm asking, because I never tried hemp paper, is it almost the same, or there are also disadvantages with this kind of
No,
paper?
it's this paper is almost same. The problem right now, the technological for the making, because conventional paper we have like big industries, and for making hemp paper right now we don't have a technology same like a conventional paper. So people are working over here to make hemp paper with the machines.
So, but your calculation are based on your very artisanal activity in India. So it's possible that with a more industrial production, calculations are different.
Different. Right now, the technological is very less in the production of hemp paper. But no one is did this one, and I take it this opportunity to calculate and compare with the conventional tree paper.
Yes, because so the question is, if this the use of hemp paper is so advantageous, why we still continue to use wood paper? I don't think
because
in general.
Because for making a hemp paper we need very less chemicals. Only bleaching we can do with the hydrogen peroxide, and we don't need to cut any trees for making. because of cutting trees, a lot of deforestation and fragmentation of natural habitats. So it can be grow with the cultivation of 4 to 5 months. 5 months. So this is the advantage.
And so why is not so popular? Why hemp paper?
Because of, yeah, because of the restrictions. Because of the restrictions. Because of it makes
hemp paper. Do you think this is the main reason for this?
Yeah, this is the main reason for this, because till 90s it is totally hemp was the primary source for paper manufacturing, and after the restrictions it totally disappeared from a market, and that's why the technological application is not very modern right now. So now slowly it's legalized, and the technological application is improving, and I think like next 4 to 5 years it will become like same conventional paper.
But what is the content of THC in this kind of a hemp that you have checked?
It's a very high THC, it's more than 20%.
So it's a drug. Basically, it's a drug.
Yeah, basically it's a drug. Yeah.
Okay.
Yeah.
Okay, thank you.
Thank you, my pleasure.
Okay, now it's the time for the second examiner, that is Professor Mucifora. Please, Simona, it's your time for shooting questions to the student. Simona?
Sì, mi sentite?
Yes.
What is cellulose chemically?
Cellulose is a compound which is present in stems, which is a very like bonding structure, polysaccharide, which used for the like paper manufacturing.
And what is GSM?
GSM is a gram square meter. Is the thickness of a paper. Gram square meter. The thickness of a paper.
Okay. Thank you.
My pleasure.
Okay, thank you very much to Rohit. And now it's time for the second student to be presented, Mr. Davide De Rossi, that I would like to see with the webcam working. Please, Davide. Yeah.
Mi sentite?
So, Yes, Mr. De Rossi will present a thesis entitled Monitoring of the Levels of Terpenes and Cannabinoids in Cannabis Plants During Drying and Curing. I am the tutor for the student. Examiners will be Professor Bellani and Professor Fabrizio Monaci. So it's your time, Davide, for reading the PDF file. Do you have it? You don't? So I'm going to show you. Wait. Can you see the writing? Davide?
Sì, sì. The Undesigned Davide De Rossi, matricola number 085616, enrolled in the course of Biodiversity and Environmental Health, considering the degree number 416/2020 protocol, team in Siena for the final exam, graduation and public proclamation, being aware that according to the Article 76 of DPR 445/2000, false declaration, falsity in public documents and use of false document punished according to the penal code, declare that under responsibility that she has participated in the graduation exam on date 16/04/2021 during the exam, during the final exam of during the degree, the exam of the academic 2019, the achievement of final qualification, that she accepts the evaluation of the final mark with which she is declared dottoressa, dottore, dottore magistrale in biodiversity and environmental health, biology,
Master degree in biology.
master degree in biology, curriculum in biodiversity and environmental health.
Okay, thank you, Davide. So would you like to share your laptop screen, please, and start with the presentation of your thesis project?
Can you see it?
Full screen. Yeah, we see your screen, but you have to activate the PowerPoint sign for full screen. Yes, correct. Keep going.
Okay, perfect. So, good morning at all. Today we are discussing this final dissertation thesis titled Monitoring of the levels of terpenes and cannabinoids in cannabis plants during drying and curing. The introduction, we can start briefly from where cannabis was domesticated from the human species, so up to 5,000 years ago. And it was domesticated for fiber, seeds, and multi-purpose use, also as we saw in the dissertation thesis of Rohit that was talking about the paper. And in this case, we will talk about the possibility of use as a herbal medicine and its property about terpenes and cannabinoids. In fact, the aim of the thesis is to understand if there's some relationship or dynamic changing in the potency with potency analysis, terpenes analysis, and also we were thinking, and we have done metagenomic analysis, but in order to see if it was possible to correlate with microbes. But because of we had a co-amplification of placidial DNA, we have we have focused mostly on the terpenes and cannabinoids and the phase of the harvest and the curing. So the plant is a multi-purpose or drug type. Obviously, we choose fiber type because we choose a compolti HTC in order to evaluate the different terpenes content and relate to the morphology and chemotype. Cannabis plant is mostly differentiated in all the different parts of the world. So we have from India and in the North Africa and in, for example, in Europe, but also in America. The problem of all the resin fundamentally, and so in this case, obviously of the flowers because they are the support of the resin, is the oxidation. So is UV sensible and temperature sensible, and so we will see after why the sensitivity of temperature in aging process briefly change the morphology, but the chemotype of the cannabinoids. But at the end, what the human wanted to consume is the the carboxylated cannabinoid that is the mostly active in the system, but now is briefly coming out also other formulation with CBDA and THCA, other acidic cannabinoids that are similar to the cannabinoids we found in the plant. The cannabis trichomes are the most important structure for pharmaceutical interest in the plant, and fundamentally they can be divided in trichomes of the flowers, of the stalked trichome, cystolytic trichome, bulbous trichome. Obviously, different trichomes have different function, and different part of the plant has different pests, and so she needed to be aware with different type of arms or weapon to launch against the nature that wants to eat also from human or in general big mammals that they can eat it. Talking about the glandular trichomes, we found mostly, especially in the cavity of the glandular head of the glandular trichomes, the most higher content of cannabinoids. And so, for this reason, it's important to develop some techniques to take out only the trichomes and not all the part of the plant, because there's a lot of compounds that we maybe can not want inside the phytocomplex. Phytocannabinoids are commonly derived from the CBGA, that is the precursor of all cannabinoids, and derived from a reaction of generanyl pyrophosphate with the olivetholic and the hyaluronic acid. The cannabinoids initially are formed also, as we were talking, as carboxylic acid, and they are produced in the plant for growth and defense against biotic and abiotic stress. The terpenes, as we were talking about, they are the most important present compounds inside the glandular trichome, especially in the flower. And so they show therapeutic potential. They are fundamental part of the entourage effect, and they have a lot of properties that we can talk for years probably. There's a lot of papers from Ethan Russo and a lot of because terpenes also has the same pathway in other type of plants. For example, the alpha-humulin, if you smell a beer, is something like to smell a flower of cannabis, because obviously they have the humulin, that is the terpenes that is similar to limonene, linalool, myrcene, and also if you consume the myrcene, you can increase the effects of the cannabinoid because you have terpenes in your blood. An important feature is the harvest time. You can see that trichomes change, mostly from clear to amber. The right moment fundamentally to harvest the plant is from the cloudy to amber trichomes, and so when they are clear trichomes, they are in a sort of terpene phase where they develop like more little bit monoterpenes and freshly flavors. When they start to be cloudy amber, they start with sweety flavor and more consistent cannabinoid content. So the plants start to be ready to be harvested also. We use the 5 clones from the same mother of controlled THDC. We were in the portion of UGL Carter, and if you want, I have a video in the slide, so maybe later we can see if interested. And we use the clones to have homogeneity during sampling. We assume that they developed the same levels of cannabinoids and terpenes because they were the same 3 clones from the same mother. So we perform drying and curing at 20 degrees and 50 relative humidity for 15 days. They stem the flowers after 15 days, place in oven bags to name the turkey bags that are special food grade plastic for professional curing bags that could be useful for the curing at large scale, because if you're doing little scale, maybe you can do it with the jar. The process of curing was also performed here in Tuscany, but in TLS, Toscana Life Sciences, in a controlled environment at 20 degrees and 50 relative humidity. This is the video, if we will have time. The summary of the curing process is the separation, once dried, of the bud from the stem. Place in the flowers and on airtight container, glass jar, oven bags, and plastic barrel, almost used for containers. And these should be filled up to 75% of their capacity, because leaving some air in the top will do a sort of space for evaporation of the terpenes and water that create a sort of little jar pressure inside that change and homogenize the compounds inside the different flowers that maybe could be a little differently grown in the plant, simply. During the first week, is important to open the jar and close in order that the flowers breathe and reintegrate the oxygen, and so release the moisture in excess. After 1 week, 15 days maximum, depending from the flowers, you can stop it and start to close and left for years or months. We choose 2 months to start the study, and when it will contain 8-10% moisture, after maximum 2 months, it's perfectly dry and perfectly soft at the same time. For different window of sampling, so at the peak of the flowering with the fresh plant, at the end of the drying, completely dried plant, during curing process at the end of the curing. For each clone, for each phase, there's not a big differences in color of the flowers, because almost if you wanted to see a big changing, you have to wait almost 6 months, because the chlorophyll will be completely degraded. The drying process was performed in Kupi, and with the upside down head of the plant in a traditional way, with the leaves, with a slow process at 20 degrees, 50 relative humidity. Could be done, for example, in aluminum trays at the same temperature in a pharmaceutical grade chamber. About the curing process, as we were talking, it's obvious that as for cheese, wine, and a lot of different varieties of food, or what, also for the essential oils, it's obvious we need to cure the compounds to obtain a homogeneous and higher quality final products. So curing is an asset of preservation techniques that aim at the retention and balance of the desirable compounds and removal of the balancing undesirable compounds. So this is possible to perform with the flowers or with the resin. And with the resin is the most interesting things in the last years in order to understand what happened, the touch, taking out the head of the trichome. So to cure means also to preserve the life of the bud of the resin, because you slow down the drying process and very, very slowly you give to the plant a sort of consistency that need to be perfect for fundamentally the consumption, but also for extraction. So you have less waxes, less chlorophyll, etc., etc. Obviously, could be involved a lot of bacteria that we wanted to sequence, but it will be maybe next work. Potential cannabinoids was performed with HPLC-UV thanks to Ambra research. Potency analysis showed fundamentally the terpene profile of CBD 20:1 ratio hemp strain. So mostly 90% was CBDA and a little part, like 5% and 1% was CBD and THC. The ratio changed interestingly from phase A to phase B and till D. So from phase A, when the plant is fresh, till the end, you see that THC over THCA over THC starts from 6 till 3. The CBD start from 17 till 11. So it's obvious that THCA is more sensible to temperature and in general aging, UV, and the CBDA is more heat resistant, so probably don't deteriorate very quickly in 2 months. Interesting. Okay. And if we see in this slide also visually what we were thinking 1 second before. Terpenes analysis was performed with gas chromatography by the laboratory of the National Center of Research with Billy Federico. The terpenoids were analyzed from frozen leaves, and the relative amount of each compound was expressed as percentage of the total terpenoids, so terpene profiles. We not evaluated the level, the potency level of the terpenes. Differently from cannabinoids, yes, we evaluated as shown. The most of the heaviest and the temperature-resistant terpenes remains at the same level, so alpha-pinene, myrcene, limonene, caryophyllene. Statistically, interesting pattern we found only for gamma-elemine and selene-ADN. Gamma-elemine was lost during drying and curing for almost 50%. Selene-ADN differently almost replicate from fresh plant to the final product. Interesting is that, for example, gamma-elemine was shown to be also anti-tumoral agent. What I think is that maybe in the fresh plants could be possible to find a plethora of compounds that we are not analyzing that could have a strong potential, and maybe it's like 100 years that humans don't analyze compounds coming from this source. This was the different lot of the terpenes, the gamma-elemine and the selene-ADN, as we discussed, they have done this type of changing, and we see here in a more specific slide the gamma-elemine and the selene-ADN, and it's possible, could be possible maybe that the increase of selene-ADN could be related to the degradation of another terpenes that degrade maybe in selene-ADN. This is needed to be shown by a more deeply analysis with mass spectrometry. In this work, we demonstrate that cannabinoids are very stable if dried and cured properly, and they change mainly the ratio between acidic form and neutral form and the content of THC and CBD. Main terpenes shown to maintain similar concentration are pinene, caryophyllene, limonene, and myrcene during drying and curing. Optical condition don't deteriorate the terpenes profile, and as expected, there is a relative increasing in certain terpenes, but selena DNA is a case, but also as we see, is shown a sort of also increase in limonene or in humulin or in bisabolin or in caryophyllene. But what we need is maybe to have a very precise quantification of the terpenes in order to be more conclusive in the data. And maybe we use indoor selection in order to be more homogeneous and maybe have higher content in order to see what could happen in the bah, in the indoor. Relative increase of certain terpenes could be the reason why cured cannabis flowers have a better taste and beneficial effects. And what we are thinking is that in general plants are expressing a sort of relationship with life, not only with compounds. And so we are thinking to do more research to understand isolating resin, what can happen with microbes and the effects on maybe cells or something also more complex like human, if we were possible. Thank you.
Thank you, Davide. And now we have space for questions. So I would like to introduce Professor Lorenza Bellani as first examiner. Please, Lorenza, it's your time.
Yes, thank you, Antonio. Okay, Davide, it was interesting to hear your discussion, and I have some questions about hemp. First of all, it was curious to me to ask you, why did you choose this variety? Because I've been working with cannabis, some experiment with hemp, and it was very difficult to choose the variety to work on. So I just wonder why you choose this one, and how much is the content in TCH, and please.
Fundamentally, if you need to choose a variety to find fundamentally the content in terpenes and cannabinoids, you need to select, for example, from seeds. And so you can find one, two, three plants that will have that characteristics. And so obviously we choose the composite because it's a plant that responds very well here in Tuscany. But mostly inside that strain, we found a mother plant that we maintained for 2-3 months to do the experiment, and from where we gutted the clones. And the THC was almost 0.4, 0.5, and the CBD from 14 to 15.
Okay, so you did also this sort of experiment in other varieties, or just in this one for the moment?
Of these terpenes we have done in this variety for the moment. But
Okay.
Okay. In this work, generally, because the problem right now is cost of the analysis of the terpenes, no possibility to find someone that could do the quantification effectively,
Okay.
because a lot of laboratories do the terpenes profiles, but we are not able to find a lot of laboratories with the quantification.
Okay, then another question about the technical aspect. You say that you have done a foliar spray of mycorrhizae, and I just wonder why foliar spray, which is the advantage?
Fundamentally, the advantage is that the plants have fundamentally a microbiome in the leaf and in all the part of the plant. So putting other type of microbes, you do a sort of selective pressure on the other population, and actually the population that are in that specific leaves, or for example in the roots, start to change, or for example, what is used, the product I used in the thesis, it was for the pest. So, for example, plants start to metabolize some secondary metabolites that take out the pest, or fungi, different type of disease.
Yes, okay, because usually,
Discorso lunghissimo.
sorry?
It's a long interview if we want to discuss only about the micros. I can summarize that generally the mycorrhizae have a strong effects on the population of microbes of the plant, and the plant start to have a metabolic change, and
Yes, but usually in the root, in the soil, because the foliar spray is controversial. I just wonder if you had information about this particular compound that you used, because maybe it was added with some other molecules that can that can have an improving effect. This was just my question. And okay, and then the last question, sorry, but I was interested, so I want to ask you something. The last question is about the secondary metabolite that you found. And actually, you pointed out that senalidiane was the one that brought in the significant increase. And the way, the moment of the collection was during the last type of collection, while at the same time you had the other one that decreased strongly. So I just wonder how you can cope to choose the right moment of treatment if at the same time one is increasing and the other one is decreasing so strongly.
Yeah, fundamentally, cioè, in my opinion, one that is decreasing or changing, in general, we have this is an indicator, in my opinion, of a strong change. We have an opposite results from our analysis, in my opinion. But if maybe we would be able to find some other monoterpenes, maybe it was possible to better understand the dynamic change. Because from my point of view, it's a little bit reductive to understand from only two statistically dependent data. Because
Yeah.
as we were talking with Nicola, maybe the gamma, the semina DNA could be a degradation of other compounds that maybe they change in that terpene.
Maybe
That is more happy.
okay. Thank you.
So,
Yes, now it's time for questions for the same examiner, Professor Monaci, please.
Okay, thank you. Thank you for presenting your data, your work. First of all, my question is about potency. Why using this terminology, potency?
what type of terminology would you use?
Content, concentration. Probably you had them. You did your analysis. If I remember quite well, you look like as a very good students in instrumental and technical parts. So I'm pretty sure you did your analysis. you did the quantification of a substance. So why use hypothesis? Is the terminology which is usually
Probably it was only a slang.
a
It was
slang, no?
sort of the American slang, differently to quantification. That
No,
maybe was
usually potency is used in dose response assessment.
okay.
So you need to assess an effect by a substance. In your case, you are determining the concentration of your substance. So that's why I was asking, because probably I thought that there was something that I didn't know. Just the only
Probably
way
was
I was asking you,
yeah,
you were continuing using that terminology. So I was, and why you expressed your data as a percentage? And why not as a mass in for mass or mass per volume?
because it's in calculated in dry weight.
So this is expressed for with respect to the total mass of the sample, if I understand well.
I explain you. Here is in dry weight
Okay.
percentage. All the pins, okay, yeah, because it's when you analyze the hemp, usually you have to dehydrate completely. The potency is calculated on that
So, I
amount.
mean, it's a quite a large quantity of this material within this sample that you analyze. I mean, 20% of some of your compounds, it's a big quantity, I think, with respect to the overall mass of carbon and nitrogen, all that stuff that is making all the biomass of this value. Is
Yeah,
okay?
in that
Is it
sense, for
understood?
sure.
Okay. Why did you express also the variance of data in the terpenes, but not in the other type of analysis with the HPLC? Because that would have been interesting to see eventual statistical differences between the different phases.
You mean in the presentation or in the thesis directly?
Both, probably.
Okay, because I think in the attackment 1 and 2 of the thesis there was some data also for
Okay.
the cannabinoids.
Okay.
I'm pretty sure.
Just to understand if those trends are significant. I mean, there is an increase of the THC, the other compounds that you were analyzing. That was a significant aspect because you used the statistics for the terpenes, but not for the other type of analysis. That's why I wanted to ask you.
Yeah, I can show you. I have
No,
thousands of
it's not so important. If you say
comparison.
that, okay, it's perfect. It's okay. Okay, thank you. That's all for me.
Thank you.
Okay, thank you, Davide, and thank you to the examiners. Now I am introducing the very last students, Mr. Cresti Andrea. That is defending a thesis entitled Effects and Translocation of Different Size Polystyrene Particles in Seedlings of Chicoryum endivia, variety Richa Cuordoro. The supervisor is Professor Bellani. The examiners will be Professor Loppi and Monaci. Before starting with the defense, I'm going to ask you, Andrea, to read the statement as your fellow student. Do you have it in your hands?
Yes, but in Italian.
All right, so I'm gonna share the screen.
Okay.
Well, let's read it in Italian. I think it is the same document, so we don't mind this.
Ok. Il sottoscritto Andrea Cresti, matricola numero 089665, iscritto al corso di laurea magistrale in biologia, curriculum Biodiversity and Environmental Health, visto il decreto del rettore dell'Università degli Studi di Siena, repertorio numero 461-2020, protocollo numero 52533 del giorno 11 marzo 2020, che autorizza la modalità di svolgimento a distanza della prova finale e della proclamazione, consapevole delle sanzioni penali richiamate dall'articolo 76 del DPR 28 dicembre 2000, numero 445, in caso di dichiarazioni mendaci e di formazione o uso di atti falsi. Dichiara di aver partecipato alla seduta di laurea del giorno 16 aprile 2021 della sessione di aprile dell'anno accademico 2019-2020 per il conseguimento del titolo finale e prende atto della valutazione e della votazione finale con le quali è stato proclamato dottore magistrale in biologia. Montefollonico 16 aprile 2021.
Okay, so Andrea, it's your time for the presentation. So share the screen, please. Full screen? Yes.
Okay, vedete?
Correct. Yes, keep going.
Okay. So, my study, I studied the effect and translocation of different sites, polystyrene particle in seedling of Cichorium mendivia var Ricciapoldogro. One of the most harmful and dangerous type of pollution for the environment is contamination from plastics. The use of plastics in the industrial field and also in our everyday life have an increase since 50 years. And from this time, year after year, we have greater discharging in all the system of the environment, like water, land, and air. And plastics not only go in one system, but can also freely migrate from one system to another. In environment, plastics can be found in various forms, in various sites. We have, for example, the more easily to observe with our eyes microplastics that have size bigger than 5 millimeters. And then these microplastics can persist in the environment and undergo to fragmentation in smaller pieces, like micro and nanoplastics. Microplastics have a size between 5 millimeter and 100 nanometer and are grouped in primary microplastics that are particle intentionally made and crafted with this size for example, for cosmetic product or also industrial product. And then we have the secondary microplastics that came from the fragmentation of larger piece of plastics through various processes like photodegradation or biodegradation or also other weathering processes. And from microplastic, from the fragmentation of microplastics, we can obtain nanoplastics that have a size between 100 nanometer to 1 nanometer. So particle that we found in the environment can be directly ingested or inhalated by animal, aquatic or terrestrial, and can be absorbed by the plant, and in this way can enter directly in the food chain and causing problem for human and also animal health. For example, for plastics can may affect all the physiology of the plant or can change the nutritional property of the edible part of the plant and so the whole ecosystem. So in this study, we use a particular kind of plastics polymer, in particular polystyrene. Polystyrene is one of the most commonly used kind of plastics that is used for electronic compound, like for TV or computer pieces, or for food packaging, and also for the isolation of buildings and also for the isolation of water pipes. And in this way, for this reason, polystyrene is commonly found in the whole ecosystem. So we study the effect of polystyrene in one specific kind of plant, of edible plant, in particular Cichorium endivia var richaquordoro, that is a common edible plant very used in human diet because of various beneficial effects, for example, antibacterial, antidiabetic, antioxidant, or hepatoprotective, and anti-inflammatory effect. And another important thing to say is that for this plant, we didn't eat only a part or the fruit, but we eat all the plant. So all the plant is edible. Then the object of the study is initially to characterize the polystyrene particle that we use in this study. So first, we analyze at TEM, at the transmission electron microscope. We analyze the shape and the size of this particle. And then with the use of FTIR, we analyze the composition of this particle. And then we subministrate to Cichorium endivia, we observe the effect of various concentration of nano and submicroparticle on Cichorium. And we observe the germination percentage, the root and shoot elongation, so the growth of the plant, and also the production of secondary metabolite like phytochemical compound and also antioxidant activity. And then we observe the production of photosynthetic pigments and also the uptake and the translocation of fluorescent polystyrene particle in seedling. So, first, we analyze with TEM the polystyrene particle, and we obtain various images. We analyze these images, and we can observe that polystyrene particle, nano and submicroparticle have a regular round shape, and in this way we can easily measure the size of this particle, and we make frequency classes with all the measurement that we do. So we can observe that for nanoparticle we have the most representative class, frequency class is the 16 to 20, and for submicroparticle the most representative classes is 191 to 200. Then, with the help using this instrument, IR, FTIR, we observe the composition, we analyze the composition of our particle, and we obtain two different, two images with the spectrum of our particle. And then we compare these two spectrum with the classic spectrum of polystyrene that we can find in the library. And we observe that the spike, the events that we found can be related to the classic spike of the polystyrene spectrum, so caused by the vibration of aromatic ring or the vibration of carbon hydrogen, and also by the CC skeletal vibration. Then we put chicolum seeds in a petri dish, and in each petri dish we put 1 milliliter of solution of control, 1 gram per liter, 0.1 gram per liter, and 0.01 gram per liter. And then we observe the germination percentage at 2 times, 7 days and 14 days, and we can observe that at 7 days we have a linear trend between control and all the concentration, for the exception of 1 gram per liter that is lower, have a lower germination percentage than control and the other concentration. But this trend is stable in 14 days. Then we observe the seedling growth, so root and shoot elongation, and we can observe that for shoot we have a stable trend between control and the other concentration, and the only exception is between control and 1 gram per liter of nanoparticle. This for 7 days. For 14 days, we have like germination percentage, we have a linear trend. And similar things we can say about shoot length, in which we have a linear trend, a similar trend between control and the concentration, with the only exception of 1 gram per liter of nanoparticle. And with at 2 weeks we have not significant difference between the control and the other concentration. Then we study the polyphenol content, and we can observe that for at 7 days we have not significant difference between control and the 1 gram per liter concentration, but we have an increase of polyphenols content for 0.1 and 0.01 gram per liter of nano and submicroparticle. And this for 7 days. In 14 days, we observe that compared to the control, we have an increase of polyphenols content in the other concentration. And a similar thing we can say about flavonoids, in which we can observe a stable trend between control and the 1 gram per liter, and difference between control and the 0.01 and 0.1 gram per liter of the two particle. This for 7 days, and in 14 days we observe that we have an increase of flavonoids content between control and the other treatment. And then we observe the antioxidant activity by using the BPH, and we can observe that between the control and all the treatment, in the first week we have a lower antioxidant activity, with the only exception of 1 gram per liter of nanoparticle. But we observe a decrease of this this antioxidant activity, mainly because the presence of plastics, of this plastic, cause loss of the activity, the power of the antioxidant activity of plant in response to the presence of plastics, and this trend is stable for 14 days. So we can we can hypothesize that in the first day the plant go in difficulty with the presence of this plastic, but in 14 days we have a response of the plant to the presence of this plastic. And a similar thing we can say for the presence of photochemical pigment. In fact, here between control and all the other treatment, we have an increase of, in this case, chlorophyll, and in the next slide we have carotenoids, in which this pigment increase between control and all the other treatment. And we can hypothesize here, also in carotenoids, we can hypothesize that we have a compensation of the plant or to the stress caused by the presence of plastics. In fact, also in other studies, in other research, we have the analysis of the response of the plant also to other studies. And this response is the increase of production of pigments. And so we can say that we can hypothesize that this increase of pigments is caused by the presence of the stress of plastics. Then we observe at light and fluorescent microscope. Here we have the image taken by with light microscope, and we can observe that in the first image we have a root apex with the meristematic part of the root, and so a newborn part of the root. And in the second image, B, we have a more older part of the root, in which we have well-developed vascular bundle and also root hair. Root hair that isn't found in the meristematic part of the cell, of the root, so in the apex. And here we have an hypocotyle. In the last image, we have an hypocotyle with a well-developed vascular system, vascular bundle. Here we have an image taken with a fluorescence microscope, and we can observe in the first image a root hair of a treated root, in which we have the presence of fluorescent light caused by the presence of nanoparticle, and we can hypothesize that by entering this root hair, the nanoparticle can enter in the root. And in fact, this thing can be proved, can be confirmed with the second image, in which we have like for the light microscope, we have an image of a root apex, in which we have the meristematic part of the root, in which we didn't have any root hair, and in fact we didn't have a fluorescent light caused by the presence of nanoparticle. And in the lower part of the image, we have a more older part of the root, in which we have root hair, and then the presence of nanoparticle that cause fluorescent light. And here in the last image F, we can observe a root with a vascular bundle well fluorescent. This caused by the passage, the presence of fluorescent nanoparticle in this vascular bundle, and for this reason is well well flourished, well observed. So then here we have the division, the passage from root to hypocotyl, and the hypocotyl is the part that we have between root and cotyledons. Here we have that at 7 days, even for the nanoparticle, we didn't have the passage between root and hypocotyl. And the second image, we have that at 14 days, we have this passage of nanoparticle even in the hypocotyl. And here we can observe various vascular bundles with fluorescent light. And this thing we can hypothesize that this phenomenon happened because with the second 7 days of life of the seed, we have the start of photosynthesis, and then we have the transpiration stream that driven up the flow of nanoparticle from root even to hypocotyl. And then here we have a photo of submicroparticle, and we can observe that in the first image we have a weakly fluorescence compared to nanoparticle at 7 days. Here we have some vascular bundle with a low fluorescent light, but some submicroparticle enter the root. And here in the last image we have root at 14 days, and we can observe that submicroparticle remain accumulated are accumulated near the rhizoderm or in rhizoderm, and the fluorescent light in rhizoderm is more stronger than in the internal part of the root. So, in conclusion, we can say that the germination percentage and the plant growth, the seedling growth, so root and shoot elongation, were mainly affected at highest concentration of the plastics, and that the antioxidant activity follows the trend of secondary metabolite content like polyphenols and flavonoids. And that the increase of all phytosynthetic pigments in the analyzed treatment in respect to the control is clearly a branch response to a stress induced by the presence of plastic, so by this treatment. And then that the imaging of fluorescent polystyrene particle in seedling evidenced an uptake of nanoparticle through the root hair by root hair with a migration from to the root vascular bundle, and this for 7 days. For 14 days, we have that this flow probably driven up by intense transpiration stream is come to the hypocotyl. On the contrary, for the larger submicroparticle, we observe difficulties to enter in the root and move to the conduction system. So the result of this multidisciplinary approach is very interesting and encouraging, and can be a starting point for further future investigation, maybe for the effect and the possibly internalize of other plastics, and also for other edible plants. That's all. Thanks for the attention.
Thank you, Andrea, and I'm giving the floor to Professor Loppi for his questions.
Thank you, Andrea, for your nice presentation and also for showing us something about the topic which is really up to date and also very important because of the possibilities of the transfer of plastic into the food chain. I have just 3 questions, one general, one methodological, and one about the results. Very simple question. The first one, the general question, why did you investigate polystyrene? Because mulch sheets used in agriculture are mostly made up by polyethylene or polypropylene, not by polystyrene. So what do you think could be the source, if any, of polystyrene in your lettuce? Could be water, from the water. So you argue that water may be polluted by polystyrene, or because you think that the soil is polluted by polystyrene, or whatever?
We observe the effect of polystyrene also because in this kind of study, large part of the study use polystyrene because by the factory that produce suspension with this particle work better with polystyrene. And another important thing is that polystyrene is very well is easily can be easily applied a fluorescent dyeing particle. And for this study, in which we want to observe even in with the fluorescent microscope, it's important for us to have particle very well visible even at the fluorescent microscope.
So you've used polystyrene because it's better suited for the experiment.
Because of course we observe that we have also an high concentration in the environment of polyethylene or PVC, but we see that in a very large number of study is used polystyrene because it's very well manageable by the factory.
The second questions, I see from the slides that your seedlings were very, very short, no? A few millimeters, hardly they were 2 millimeters in length.
Is with its length 3 millimeter and 1 millimeter of
the length of the shoot. One of the next slides you've shown that there are 2 millimeters, more or less. So I was just yes, this one. You see, around 2 millimeters, correct?
Yes.
So I was wondering how it was possible to collect.
Probably we have an error because it's in centimeter.
Centimeter. Otherwise, yes. The question was how it's possible to have enough material for all the analysis, chlorophylls and polyphenols.
Also because to collect the germination percentage, to say that a seed is germinated, need to have a root longer than the diameter of the seed. So this probably is an error on the graphics.
So 2 centimeters, which is anyway a small plant anyway. Okay, okay, you can leave this slide. It's clear. You see that in general, I've seen polyphenols are higher after 2 weeks compared with 7 days. Higher concentration. But you have cultivated your plants, your seeds and seedlings, the first week in the dark and the second week under the light. So, do you think that the effect can be determined by the time, the longer time, or by the light? Is it possible to disentangle the effect of time and light, for instance?
So the effect of the light and then the start of the photosynthesis can be for sure related to the increase of the pigment content, like chlorophyll. Here, for polyphenols, we have this observation, but we didn't say for sure that is because of the presence of light, or maybe because with the growth of the plant, with more time, plant can make some response to the stress, but we didn't know if is related to the presence of light, or maybe even in dark, plant can have this behavior.
Okay, thank you. Very last question. Do you think that your results can be generalized to other plastics? And is it safe for humans to feed on your cyclery?
Maybe for yes, probably also for other kind of plastics, also for other kind of polymers. But the important thing is that we can use nanoparticle. In fact, polystyrene is very easily to produce nanoparticle. So we need nanoparticle and also submicroparticle. And then for if for us it's safe to eat, maybe we need further investigation, even because we didn't have such research on this argument. So we need to say the way of this particle can reach edible part of the plant, and for also for other kind of plants, for plants that have fruits. So we have to follow the flow of nanoparticle and also observe where they go, where they are situated.
Okay, thank you.
Thank you.
Right. And it's now time for the other examiner, Professor Monaci, please.
Okay, thank you for presenting data.
Thank you.
You did very well. Also, the thesis is well done. Just one question for me is related to the germination study. You used nano particles. We were with some co-formulants. Do you think surfactants or other substances that were within the product that you used as a starting point for the experiment? Do you think these substances may have had any effects on your results? You can discuss about it.
In our study, we use only water and the suspension that we buy with nanoparticle. So we administrated to the seeds or only control or the other treatment, but only 1 milliliter of these solutions. So
okay. So no effects, nothing to okay, thank you.
So
oh, it's okay. Perfect. Thank you very much.
Thank you.
Okay, so if everything is done and finished, I'm kindly asking the students to hold on in this virtual room. Meanwhile, I am transferring all the committee members in another virtual room. Okay, maybe I can do that. Please, Andrea, disconnect your screen.
Sì, non so come sto guardando.
Red button.
bottom there is condividi.
Non so se non mi riapre. Perché mi ha messo in modalità in un piccolo riquadro e non so come tornare. Non so come.
Dunque,
Antonio, credo che se gli togli la proprietà di relatore il quadro si cancella da solo, ma non ci giocherei.
cambia ruolo. Lui è figura, ok, assegna privilegi. No.
Maybe, prova Ctrl Tab, Andrea, se ti appare qualcosa.
No, non mi sta.
Ctrl Tab, se ti gira. Maybe you can close something that you...
Prova a chiudere.
Infatti, disconnetti che poi rientra.
Ok.
Anche provo. Sì. Perché proprio mi ha messo un riquadro piccolino e c'ho soltanto 3 tasti per modificare la visualizzazione. Infatti ho fatto tutta la presentazione con questo riquadretto.
È così, disconnetti.
Ora prova a schiacciare X? No.
Però me lo chiude soltanto giù.
Vai ancora. Vai su visualizzazione dello schermo, ce l'hai, no? O è mio? Sul pulsante visualizzazione dello schermo.
Vabbè, non importa, intanto noi ci spostiamo sull'altra room. Aspettate un attimo, quindi Andrea e tutti gli altri aspetti. 1, 2, 3 De Rossi no, Rohit no, passa sessione interattiva,
Accidenti.
avvia, accedi.
Cioè, dobbiamo pigiare qui join now?
Sì.
Accedi ora.
Sì.